Hello my friends, and welcome to another video by Synic. We're going to be looking at our first biology topic here today, which is cell biology. This is the whole collection of Biology Paper 1. Anything that you come across in this video that has marked HT is higher tier only, and anything that says BI only is only in the biology for triple or separate sciences, is not in combined science. Everything else is in combined science. So what are we going to look at? Well, we're first going to look at cell structure, followed by microscopy, the things we need to know. We're then going to look at biology only part, which is culturing microorganisms. We're going to look at mitosis in cell division, stem cells and where they come from, what we can use them for, and finally transport through diffusion, osmosis and active transport. Let's go. So first up, we're going to look at cell structure. And the first definition that we need to know are the two simplest categories of life, which are prokaryotes and eukaryotes. And if we break the word down, we've got the pro and the eu, and the rest of it is the same. So pro means it has no nucleus. Simple as that. There is nothing else really that defines a prokaryote apart from its nucleus or not. The eukaryotes, they do carry a nucleus. So these are eukaryotes. Eukaryotes, and these are nokaryotes. Simple. But within those, we've got different categories of things. So in prokaryotes, we have this group of bacteria. So this is a bacterial cell here. And within eukaryotes, we have a plant cell and we have an animal cell. I've got those the wrong way around, but it doesn't really matter. They're both animals and plants. They are eukaryotic cells. But if we look in a bit more detail, we've got to be able to label some of these parts. Let's start with eukaryotes first because they're probably a little bit simpler. So these do have a nucleus, and there's our nucleus there for this first one, and the nucleus for the plant cell here is this one on the bottom right. I've drawn in some chromosomes there just to show that they are housed within the nucleus, and you'll see that there is no nucleus within the bacterial cell. However, there is a chromosome, and it's this loop of stuff here. That there is a chromosome. They have a single chromosome, and then they've also got little packets of plasmids, which are packets of DNA. They are little circles of DNA, which can also be shared between different bacteria. Quite cool. Anyway, also in our eukaryotes, we have mitochondria. You probably learned those in Key Stage 3 as the powerhouse of the cell. Well, they are the site of respiration, which is well worth learning. The vacuole is only found in plant cells, as is a cell wall, which is this layer here, and chloroplasts are usually found as being green in plant cells. We can draw those in green. Where's my green? There's my green. Draw them green. Draw them green. Green, 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 green. So those are found in plant cells only. They are what make plants look green. They are not going to be found in animal cells at all. However, in animal cells we also have, as well as in plant cells, things like the cytoplasm. And the cytoplasm is just a liquid within the cell, so that is here and here. And we've also got tiny little dots here, things called ribosomes. And the ribosomes are found in both of those cells, and they are where proteins are made. So tiny little structures, and they're really important. But those are also found in our prokaryotes. There we go, there's one little one there. So ribosomes are found in all three. Cytoplasm is found in all three as well. So it's in the bacteria, it's in the it's in our animal cell, it's also in our plant cell, so it's in all three. And you'll also notice, well actually that's our eukaryotes done, isn't it? They're all labelled up, brilliant. Whereas the cell wall is also found, it was found in our plant cell, so cell wall is this bit here, but it's also found in our prokaryote. It's a slightly different structure, it's not made of cellulose like plant cells are, so this one here is cellulose. We don't need to know what they're made of in bacteria or prokaryotes, but they still have a cell wall. I've labelled that a chromosome. They do have that one there. And they've got these packets of DNA, as we called plasmids earlier. And they also have usually a mechanism of getting around. So something like a flagella, or they might have tiny hairs on the outside of their cell called cilia. But the key one to know about is the flagellum, because that's also found strangely in sperm cells, but they're quite a common feature of prokaryotic cells like bacteria. So just to summarise, we've got prokaryotes are bacteria. They don't have a nucleus, whereas eukaryotes do have a nucleus. Key structures are the nucleus, which is the site of where DNA is found. DNA is then copied to make proteins, and the site of protein production is a ribosome. If you don't have a nucleus, you've got a chromosome, which is just free floating around, and a plasmid, which is free floating around. We've then got mitochondria, which is the site of respiration. We've got chloroplast, which is the site of photosynthesis, which contains chlorophyll. And I'd say those are the main ones, really. The only thing that I haven't labelled otherwise is the cell membrane, which is going to be this inner layer on the prokaryotes. It's this one and only layer in the animal cell, and it's this inner layer in the plant cell too. Otherwise, that's it. When we look at cells a little bit more, we realise there are some specialist cells that we need to understand, and these have all differentiated or specialised. They've gone from the traditional view of what an animal or a plant cell is, and they've become specialised for a certain function, a certain role. And we've got a group of animal ones and plant ones that we need to know about. So let's dive into them a bit more. So first up, we've got sperm cells, and these are all about reproduction, but sperm cells have to do a lot of work. They have to move quite a long way compared to their relative size. And so to do that, they've got their flagellum, which is a single flagella. It's a tail that allows them to move, it just spins around. And to enable them to spin around for a very long time, they have in this section of the sperm cell lots of mitochondria. So it's just to be aware that they might ask you to talk about that. That's the site of respiration to provide lots of energy. They've also got a little nucleus in there with a chromosome, and because they need to contain half of the DNA that a whole organism, whatever this organism that needs the sperm is from, they need to contain half the DNA of that whole organism. So when they combine and fertilize with an egg cell, that DNA will combine with the egg cell's DNA to make a whole cell DNA, and therefore go through replication to make all the rest of the cells of that body. And it also has a little pocket of enzymes here in the head of the sperm cell, which digests the outer part of the egg cell so it can get in and fertilize that egg cell. Next is the muscle cells, and they need to enable motion. So in these muscle cells, they have long fibers of protein. And these cells actually are, they were many individual cells that have actually formed a longer cell, so that they have the opportunity to have these long fibers, not just working in one small cell section, but in a very long multi-nucleated, meaning it's got many nuclei in one large, long muscle cell. Similarly, for a nerve cell, they're quite strange in that they can be very long, but this is actually just one cell. So if you imagine you've got a nerve cell that passes from your fingertips all the way to your spinal cord, that's a very long axon, which is the part of the body, part of the cell body that kind of extends outwards and connects to another nerve. I've drawn this one here just to show you that it's got these lots of spindly finger-like projections that can connect to other nerves. So this was a nerve cell that might be found in the brain, so it can have lots of different pathways across synapses, and you'll look at that more in paper 2. But they are about communication, so sending electrical messages. So they have lots of active transport happening within, as a result of that active transport, like with the sperm cells, they're going to need lots of mitochondria. Another cell that's going to need lots of mitochondria is the root hair cell, because it has a focus on transport. So these are found in plants, they're found in the plant roots, and they have a high surface area. That's their focus, really, is having a high surface area, their specialist role. The reason they're called hair cells is because they have this hair projection, and that hair projection is what is going to enable them to have a high surface area. So the root continues up and down from here, and this finger-like projection of hairs go out into the soil around them, increases their surface area, gives them a really large surface for absorbing water or mineral ions. But the cell basically does two processes, which are the most important bits: osmosis for the movement of water and active transport for the movement of mineral ions against the concentration gradient, which we're going to revise a bit later. And the final two are for transport, which is the xylem and the phloem. Again, in plants, so they're in the vascular bundle in plants, which is where their plant veins are, if you like. The veins are the xylem and the phloem in the vascular bundle. And the xylem is for transporting water, the phloem is for transporting sugars. The xylem is key in that it is basically a hollowed out section of dead cells. So this is almost like tree bark is what xylem are, and the water can just travel up these from the roots to the leaves. It's that movement from the soil upwards. So this should be really vertical, but that's fine. Sugars travel in the phloem and travel through these cells. These cells are very much living, or they have a cell body which is attached, which is living, which sits kind of like here. And they move through these little sieve panels, but again, the sugars will travel to where they're needed, so generally they're either stored in the roots, so they're moving from the storage. It could be that it's like a potato is a store of sugar and it's moving from that, or it could be that it's going from the leaves where it's being produced to wherever it needs to. So this can move both up and down the plant, whereas the xylem is only ever going to move up the plant. So the next part of the topic is to look at actually how do we know about cells, how have we found out about them? And it goes back to the history of microscopes. Way back in about 2,000 years ago, we started developing our first lenses, and lenses allowed us to see small things slightly larger. And it came to a man called Robert Hooke a few hundred years ago who developed the first proper microscope and first saw what we now call cells. And it was the light microscope that was developed at the time that allowed us to see that. Now, in more recent years, we've developed things called electron microscopes. Light obviously uses lenses and focuses light to make things have a higher magnification. It makes things bigger, and it's allowed us to see cells and large subcellular structures like nuclei. But it's electron microscopes which fire electrons at surfaces, which allow us to see a much higher resolution and a much higher magnification. And this has allowed us to have a better image. Better resolution basically means that we can distinguish between 2 points much better than we could for light. Light has a limit to how big we can magnify things because of its resolution, and it's allowed us to see the insides of things like chloroplasts and mitochondria and ribosomes and plasmids, and it's allowed us to see those much better and therefore understand cells a lot more. Now, what we need to understand is firstly these two terms, resolution and magnification, and we need to know that for electron microscopes they are higher, so they are much better resolution, a higher resolution and a higher magnification. We also need to be able to do calculations using an image size, an actual size of a cell. The image size is what you've seen on your page, and magnification. And you'll have to work with questions using the equation here, I equals AM, or if you put it into a triangle, which I don't really agree with, but it can be useful. This is pretty much the only equation you need to know for biology. You've got that here. So image is the image size, so you'd measure that using a diagram, using a ruler. So in your exam paper you'd have the image size, you actually measure that, usually best to do in millimeters. The actual size is the actual size of the cell or the structure or the thing that you're trying to measure. Magnification is how much magnified that image is compared to the actual object. The only thing that you must remember to do is to convert your measurements to the same unit. The best way to do that is to convert either the micrometers that you're probably given for the cell size into millimeters, or vice versa. So if you have an image that an actual size that's 10 micrometers, in millimeters you need to divide that by 1,000, so you get 0.01 millimeters. If you're going the other way, so that's times them by 1,000. If you're going the other way, you're dividing, sorry, that's the wrong way around. You're dividing by 1,000. And if you're doing it the other way, you are multiplying by 1,000. And the reason it's better to work in millimeters rather than centimeters is because otherwise you're working in tens of thousands, and it just gets a little bit more complicated. So measure in millimeters, convert it to the same unit, put it into this equation. Job is one that's good, nice and simple. In terms of this required practical though, what you need to have to do is observe, draw and label a selection of plant and animal cells. The key things to note and remember are things like using a stain. A stain allows us to see transparent substances better, so cells might make cell walls more visible, it might make starch more visible, it might make DNA more visible. The key thing is it's making it more obvious. We would obviously then use transparent glass slides which allow light to pass through, and a very thin layer of tissue like 1 cell thick epidermal tissue from onion, or cheek cells. They are the standard ones. They're thin enough, small enough for light to pass through. When it comes to drawing them, you should use a clear, unbroken line, and we should label using a ruler, using straight lines, and using a ruler, and they should be going straight to the actual item, so straight to the nucleus. No gaps in between where your arrow ends and the actual item itself. It must be touching the edge of the surface of the thing you're trying to label. And we shouldn't really cross over our label lines either. In biology only, we have an extra section which is a required practical, which is where we have to know how to culture microorganisms, specifically bacteria. And bacteria reproduce really quickly using a process called binary fission. And it's a process here where we've got one cell which elongates, grows longer and longer and longer, and eventually we have this budding happening in the middle. It's budding in yeast cells, it's called, but it's binary fission in bacteria, where they basically pinch off the cell membrane and the cytoplasm, which gives them two separate cells. So they've had to reproduce the structures within the cell, make more ribosomes and make more copies of DNA and make more cell membrane, so that when they've divided, they've then got equal amounts of both in each cell, all the things that they need to survive. So they need to make sure that they've got enough nutrients to do this, and they also need to make sure they've got a suitable temperature to do this process too. And so if we're trying to culture these organisms, we need to give them the right nutrients and we need to give them a suitable temperature. So what we need to have agar jelly, agar nutrient jelly, which has the nutrients that you need to be able to grow, and we need to give them a suitable temperature, which we'll talk about in a minute. So first off, when you're doing this, you need to make sure that the surfaces you're using and the materials you're using are sterile. That means that they've been heated to a high temperature, or if they're in a more sterile place like a laboratory in a hospital, these would be done using radiation to kill everything else that's on them, and they'd be kept closed and sealed in a bag or something like that. So the petri dish must be sterile. The culture media must be sterile, apart from the actual culture that we want to grow and replicate. So if you're given E. coli, for example, that E. coli container should be sterile apart from the E. coli in it, and the inoculating loop that we're going to be using should be sterile as well. And that inoculating loop will be used to inoculate our petri dish. So in our petri dish, you would have your nutrient agar, and you'd be streaking it along different directions to encourage growth inside it. Sometimes you might be given a petri dish that's already got agar jelly in it, and it might already be seeded with bacteria, which means that it's already equally spread across the surface of the agar, so it will just grow evenly across it. Once you've done your test, be it putting antibiotic discs on it or tested for different types of quality of soap, you would then secure the petri dish with adhesive tape, but not sealing it. The reason we don't seal it is because if we did seal it, we'd encourage anaerobic bacteria to be growing, and anaerobic bacteria would be potentially pathogenic. And pathogenic means that they could cause disease to humans. So we don't want that to happen, because these anaerobic pathogens are the ones that would cause infections in open wounds and things like that, and they're generally pretty nasty. Okay, we'd then secure it upside down as well, just to prevent condensation from falling onto the media that we're growing, onto the culture. And we'd also then incubate it at 25 degrees, which is a high enough temperature to encourage growth. So it encourages quick growth, but it also prevents pathogenic bacteria growing again, because humans live with a body temperature of about 37 degrees, and if we had a temperature close enough to that, it would encourage those, if we grew it in a temperature close enough to that, they would encourage pathogens to grow. So we want to have it at a low temperature, but high enough to encourage the growth to happen quickly. For our experiments using things like antibiotics, we've got a disc here, got another disc here, got another disc here, and another one. If we put it onto bacteria and we let the bacteria grow, you would start to see these zones of inhibition, where you would see these clear areas around these discs. And these basically are assigned to say how much the antibiotic is preventing the bacteria from growing. And what we need to do is we need to take a cross-sectional area of that colony, and work out how large that area is, and compare these to find out which is the most effective antibiotic. So what you would do is you would find out a bunch of diameters. So you'd take a diameter at many different angles and take an average of all of those, and that would give you the average diameter, and you could use that to work out the area by converting it into radius and then using pi r squared. Some of them that are more like circles are a bit easier to do, but the general idea is to take many different readings of the diameter of the zone of inhibition, so that we can take an average and it be a mean average, and it's much more accurate as an outcome. So that's how bacteria replicate, but our cells work in a slightly different way. They go through something called mitosis, And mitosis is our version of eukaryotic cell division, where we make two genetically identical cells. So they're genetically identical. They may not look the same necessarily, but they are genetically identical. That means the amount of DNA that they have, the full set of DNA they had before, ends up being in both the daughter cells that are produced at the end. Now, we do need to know some of the stages involved within it. We don't need to know loads about it, but we do need to know some of the stages. And that is, firstly, that the genetic material doubles. So you can see here that we've got a chromosome, and it is making a copy of itself. So this is where it looks like an X chromosome. It actually has two halves to it. So this is one complete set of DNA, and this is a copy of it. And every single chromosome goes through that replication, so it can then be split down the middle. And each set of that DNA can go to each of the new cells. So it ends up being here. We've got one, we've got two. That's a full chromosome each. So the genetic material doubles. We also have to have an increase in the number of subcellular structures. Now that's the things inside the cell, like the ribosomes, the mitochondria, and we've said the DNA replicates. So that's just so that the cell can grow, just so that it can then divide in half and it can have everything that it needs. Of course, that's not literal split in half because the DNA needs to split too, but so that it has enough mitochondria to go to each of the new cells that are produced. The cell then divides into two identical cells, and to do that, one set of chromosomes is pulled to each end of the of the cell. So if you imagine here, we have our chromosomes divided, 4 on the bottom and 4 on the top. And this is the actual part of the mitosis where the nucleus divides. That's what mitosis actually is called. So mitosis is part of the process of cell division. Finally, the cytoplasm and the cell membranes will divide. They'll split in half. The cell membranes will pinch off to divide to give two new identical cells. And this is really necessary for us, for our growth and our development. So our cells replicating, making more of them. So you will go in your lifetime from being just one cell that you started as, fertilized embryo, going to form the hundreds of trillions of cells that you are as a fully grown adult. And the other thing that we need it for is for repair. So where we've had damage in our bodies, we use mitosis to replicate the cells to repair those. The majority of the cells that are undergoing mitosis in our bodies are the stem cells, and the only cells in plants that are going through mitosis are the stem cells. And these cells are undifferentiated cells, which can give rise to many more cells of the same type. So if they're fully undifferentiated, like embryonic cells, then they will make many more fully undifferentiated cells like embryos. If they are bone marrow cells, they're going to make more bone marrow cells. These stem cells, once copied, will give rise to other types of cells, so other types of cells from a process called differentiation. And differentiation is where genes are turned on or off to allow it to specialize, and it will make certain features, like the specialized cells that we saw earlier. It might make a flagellum for a sperm cell, it might remove a nucleus for a red blood cell, it might make cilia for hairs, for the cells in your throat. All have different roles, but the types of stem cell that we find, we have three categories. In humans, we find two types of stem cell. One is from adults, which is found in their bone marrow, and this is common. We can donate bone marrow, and bone marrow is used to form many types of cells, including blood cells, but usually it is just blood types of cell, white blood cells and red blood cells, for example. And these are stem cells, but they're already quite specialized. So if you imagine that specialization is a bit of a spectrum, where you could have fully undifferentiated at one end of the spectrum, and you've got differentiated, as in these cells cannot differentiate anymore, is over at this end. You might find like red blood cells are over at this far end here. They cannot specialize anymore, whereas human bone marrow cells might be somewhere about here between the two. They are still called undifferentiated, but they can go on this pathway to differentiation to become white blood cells or red blood cells, for example. Human embryo cells, though, however, can be made using things like therapeutic cloning, or can just be embryos themselves. These are fully undifferentiated, and this means that they can be cloned to differentiate into most of the different types of human cells. It depends at the stage of what stage this embryo is in, but most of them will be pretty much fully undifferentiated, so they could become any type of cell. So if we wanted to treat diabetes using stem cells to make new pancreatic cells, we can do that. So these are undifferentiated, very undifferentiated. They can become many different types of cell, many cell types. And the final category here is meristem cells, and these are the only parts of plants that will be doing replication, the only parts that will be replicating and therefore can differentiate. The rest of the cells, if they're going through, if the plant seems to be growing, it'll be either cells are being replicated in the meristem, or it's the cells themselves that have already been made that are growing. They're literally elongating, they're getting bigger. And the meristem is found in different parts of the plant, usually the bottom of buds and leaves and branches, and these are where mitosis is happening. But this tissue can differentiate into any type of cell. So you can take a cutting that includes a meristem, and you can form a full plant from it, because the cells will replicate and they'll form a full part. So you could take a leaf, you can pull it off, as long as you've got the bottom part of the leaf where it's attached to the branch, and you can potentially plant that and it will potentially grow into a whole new plant. It's called a cutting. So these are also fully undifferentiated. And therapeutic cloning is where we are taking an embryo and it's produced with the same genes as a patient. And this basically means that we're able to produce stem cells which have exactly the same genetics as the patient, and therefore these stem cells that are put into the patient are not rejected by the patient's immune system because they are genetically identical. So they're used in medical treatment. However, there are many potential risks of doing this. So, for example, it could cause a transfer of viral infection. There are also ethical and religious objections against it. So the fact that you're using cloning, you're producing embryos, these embryos have the potential to go and form a full life. And by using these embryos in stem cells in people and in research, those embryos can get destroyed. And therefore you're potentially ceasing a life from being able to exist. The stem cells from plants, the meristems, can also be used, so we can take these stem cells and we can quickly produce clones of them, either using agar or by taking cuttings. And we can, for example, protect rare species from extinction, so we can make many copies of them and then release them into the wild. And we can also crop plants, use cloning here to make many of the same type of crop plants which have special features, for example disease resistance, maybe making loads of copies of the same type of banana or maize or whatever it is, so get loads of copies for that group of farmers. Okay, the final section of this topic is looking at how we transport things around cells and between cells and in plants and animals themselves. And we use three different methods. The first is one which is not needing energy at all, just happens completely naturally, doesn't have to happen across the membrane, is diffusion, which is the net, which is another way of saying overall movement of things from a high concentration to a low concentration. So starting at a high concentration here, and it's just moving down a concentration gradient to an area of low concentration. And that does not need energy because it just can do it freely itself, pushed around because of other particles colliding with it, and it's just going to cause it to randomly move to a location where there's a lower concentration. Simple. Osmosis is still diffusion, but it's the diffusion of water, and the phrasing that we use to describe it is slightly different. We phrase it as the movement of water from a dilute solution, so where there is technically a high concentration of water, to a concentrated solution, so where there is technically lower amount of water. So it's still diffusion, it's still going down a concentration gradient, it still doesn't need energy to happen, but it does need to have a partially permeable membrane, which is this here, where water can move across it, but the solute, molecules like sugar or salt, cannot move across that membrane. And what happens is, as a result of that, there's an imbalance. So we've got the concentrated side, and we've got the dilute side. Think of it like drinking squash. One is concentrated, one is dilute, and water will move from the dilute side to the concentrated side, to the area which is more concentrated in terms of the solution, in terms of how much solute there is. And we'll see a required practical of that in a moment. The final type of transport here is called active transport, and that goes against the concentration gradient. So we're going up the gradient, we're going against it, so going from a low concentration up to a high concentration. And this is where there's a net movement of molecules from a low to a high. Now this does require energy. It requires energy to give the push up this concentration gradient, push up the hill if you like, but talk about concentration gradient. Yeah, that's the final type of transport. So we've got many examples of diffusion that we could talk about, from things dissolved in a solution or particles of a gas, all moving from a high concentration to a low. So we could talk about oxygen and carbon dioxide in gas exchange. Those are really good examples. So in the lung or in a leaf, they're moving across the membrane or moving into the leaf itself. And we could also talk about waste products like urea moving from cells into the blood plasma for excretion later in the kidney. But all of these surfaces need to have a high concentration difference, because that increases the rate of diffusion, high conc difference. We also need to have a decent temperature, because temperature increases the kinetic energy of the particles, so it gives them a faster movement speed, and therefore increases the rate of diffusion. So in our bodies, for example, 37 degrees is great. And we need to also have a high surface area, because the higher the surface area, the more space there is for particles to move across. And so a single-celled organism, for example, has a relatively large surface area to volume ratio, which allows sufficient transport of molecules into and out of that cell to allow it to live. And we can't do that because we're so large. We have a low surface area to volume ratio, so we need to have lungs and blood to circulate the nutrients that we need in our body. We could go on to look at, and you do need to be able to talk about, surface exchange, gas exchange in examples like the small intestine and the lungs, and the roots and leaves, for example. And these are adapted for really good exchange of materials, so they have this large surface area. Temperature is kind of sorted, but they do have a high concentration difference. So for example, in your lungs you have a flow of oxygen coming in through the air, and you have a flow of carbon dioxide coming in through your blood, and there's a large concentration gradient between the two, which makes diffusion happen quickly. We also have the similar system in your gut, so you have your materials moving through your gut in a different direction to the blood that's moving up past your gut, which makes it work quickly. We also need to make sure that the pathway for diffusion is thin. Having a thin diffusion pathway makes it work quickly, because there's less distance for the molecules to move across. In animals, we've spoken about the idea of having a concentration difference maintained all the time, but having a good blood supply helps with that. And for gaseous exchange in your lungs, you need to have a good ventilation, so being well ventilated makes gas exchange happen quickly too. And the same would happen in leaves too. If it was a windy day, you'd have faster rate of transpiration, so water being dragged out of leaves, and more circulation of CO2 and oxygen around the leaf too. In terms of examples of osmosis, we can talk about osmosis in root hair cells for plants. That's a very good example. Water is moving into the area which is more concentrated, which is within the root, and that's really lovely. We've got the idea that we are pumping things into the roots, so sugars might be stored there, we might have mineral ions moving into the roots, and with that would come water through osmosis. For active transport, the examples we can talk about are sugar in your gut. So in your gut you have a low concentration of sugars, because most of it's moved out by diffusion, and in your blood outside of your gut you have a high concentration, so we're pumping sugar out of your gut into your blood up a concentration gradient, which costs energy. And in plants we have, again in root hair cells, we're taking the very dilute solutions in the soil, we're taking the mineral ions out of that, and we're pumping them into the roots. Okay, for osmosis we have a practical that we need to be able to do. This is for everybody, biology and combined science, and we've got various things that we need to be able to be able to do. The key description of the practical is to investigate the effect of changing the concentration of a solution being either a salt solution or sugar, and seeing the impact it has on plant tissues, which we can see the impact of by measuring the mass, and we can also measure the length, but mass is probably the better one to do. So what we tend to do is we measure the dry mass of the potato or the beetroot or carrot, whatever it is, before the putting in the solution, and then after a controlled period of time, and we calculate the percentage change in mass of that material to look at improving its validity, because you could look at measuring just its mass before and its mass afterwards and looking at the change, but it's likely that all the materials that you started with won't be the same mass. So if you have a potato chip that starts with 1 gram, for example, and it increases to 1.2, that's a very different change to a potato chip that started as 2 grams and changed to 2.1. The change in mass is 0.1 gram, but the percentage change in mass is much different. It's a 10% increase or a 5% increase. So percentage change in mass is a much better dependent variable to measure or to calculate. And to calculate it, what you need to do is work out your change in mass first, so what it was compared to what it was afterwards, and the difference between the two, divided by the initial mass of that plant material. If you multiply that by 100, that gives you your percentage change in mass. Simple. Other things that you need to control would be the volume of solution, but generally you just need to have enough to cover the vegetable or whatever it is that you're investigating. We do need to control the temperature, because the temperature will affect the rate of osmosis. We need to control how long it's been working for, so the time it's been going for, because more time means more diffusion of the water can happen. And the type of solution is important too. Obviously you're going to be investigating sugars or salts, but the type of solute that you have would affect the rate of osmosis as well. There are various errors that you need to check for here, one of which would be the vegetable not being fully dried. So once you've taken it out of your solution to measure its mass, if it's not fully dried, you're going to be measuring the mass of the water on the outside too. And the other thing that can happen is you can have water evaporating from the beakers or the test tubes that you're working with. So if you've left it for a couple of days to run the experiment, water will evaporate from the beaker, the test tube, the boiling tube, and it will affect the concentration. And if you're affecting the concentration, you're affecting the independent variable. It would have an impact on the results. So the best thing to do would maybe to be put a lid on it, put a lid on these beakers, put a lid on the boiling tubes or the test tubes. Or if you can't do that, then make sure that they're all exposed to the same amount of evaporation and you started with the same amount of volume of solution to begin with. But lid's probably the best way to go. Improvements you could do is having a minimum of 3 repeats, which is a fairly standard thing. If you take repeats, you can identify outliers. If you have 3 repeats as well, you can also calculate a mean percentage change in mass, having eliminated outliers before you calculate that mean. And you can also use balances which have a higher resolution. Resolution meaning how many decimal places you have. So 2 decimal place scales would be much more accurate than using a 1 decimal place scale, because it's closer reading to the true value. The final thing that we need to be aware of here for this experiment is that you would get a graph which has positive change in mass and a negative percentage change in mass, which is over here. And what you would end up doing is plotting your graph. So when you've got a 0 concentration, which is basically dilute water or pure water, distilled water, you'd have plots up here, which would slowly work their way down like this. And you'd end up with a fairly straight line, hopefully, which goes through that graph. And the point at which it overlaps that x-axis at 0 concentration, 0 change in mass, actually tells you the concentration of the potato that you have, because at that point the net movement of water is 0 because it hasn't changed mass. And so with that, for a high level question, you might be asked, how do you find out the concentration of the potato or beetroot or whatever it is, and the sugar or something within it? You can just say it's the point at which it overlaps the x-axis. It's 0, where it has 0% of change in mass, because it hasn't had a net loss or gain of water. Thank you for watching, guys. Do check out my other videos for different topics and the key skill videos as well.